Any questions? Over past material? Oh my God, you got a Ducat? Jeez.
Look. Just be a little too quick.
I hope they lose. All right, so I posted the quiz over the insect hosts. I was working on a stickosome bearing quiz, and advising appointment took a lot longer than I expected. But we'll get those up probably by Monday. Make sure you take that quiz. The quizzes, it's general and then also life cycle questions. That's due Monday night. Start reviewing the material. And so we are moving to the GI tract resin. So as I said with the nematodes, we broke it down into invertebrate host and vertebrate host, and within the vertebrate host we have our stickosome bearers, Trichinella, Trichiurus. Now we have our GI tract residents, and then we're going to wrap up with our tissue dwellers, those that are outside of our GI tract. So the GI tract, actually I broke it down into hookworms first, because we cover a lot about hookworms. All right, and then we've got like the part 2, which is the rest, which includes Ascaris, pinworm, Enterobius, and Camelinus. All right, so those are all specimens that we have in the lab. So today we're going to start with our hookworms. I don't know if we'll get through it or not. But this is the first part of our GI tract residents. So with these residents, they're going to live in the GI tract. All right, stickosome bears typically do live in the GI tract as well. They've got a little bit different life cycles because of sticosomes. The sticocytes play some sort of function in their life cycles. Now, we can think about how these life cycles evolved. How did these parasites get there? And there's actually 2 hypotheses as to how we've developed this parasitism. First one is linked to accidental ingestion of the egg or the larva. All right, accidental ingestion. So the idea here is that you have animals. I should say animals. Yeah, animals. You have organisms that, as they've migrated onto land, or during ancient history, right, they've started to increase in size. As they've increased in size, they are now increasing their food consumption because they have higher energy demand, and also increasing their diversity of food that they're consuming. Again, their nutritional requirements are increasing as they're getting larger. All right. As you have this increased food consumption, you're going to increase the chances that you accidentally consume some of these nematodes. So the nematodes evolved well before the vertebrate hosts, and we're talking about these vertebrates that have come onto land. All right, so you have accidental consumption of the worms themselves, all right, the adult worms. And then you have some of those individuals, or most of those individuals, probably killed, probably digested. But all it took was a single mutation that allowed the worm to survive and pass through it. So now you've got this mutation where the worm can survive accidental consumption and continue reproducing out in the soil. So these are, soil microbes, soil nematodes. But what if another mutation developed that allowed the worm to persist just a little bit longer? And because they persisted just a little bit longer, now maybe they've avoided some sort of harsh environment, or maybe being in that host allowed the worms to get to a new area and colonize a new area. Well, now that mutation has higher fitness and will be selected for in our population. And you start getting this cascade of selection up to the point where now parasitism becomes required. So the worm is in the gut, it needs to get into the gut, and then all of a sudden maybe it says, well, let's not leave at all, let's let our eggs get out, and then the larvae then are the ones that need to get consumed. So, the first way in which we develop this parasitism is probably through I lost my train of thought because I read this line and it doesn't make any sense. Slowly because required, then develop parasitism. What was I trying to say there? Anyways, it's tied to increasing food consumption. We're gonna fix that one. Because again, that just bothers me. I don't know what I was saying here. So this method, I guess maybe slowly became required. Then developed parasitism. I think that's what I was trying to say. Oops. Slowly became required. So in terms of the requirement, the passage through the gut becomes required. And then at some point, it then developed the need to remain there, which would then be the parasitism. I think that describes exactly what pretty close to what I was trying to explain. All right, so that's one method. The second method involves those parasites that have, let's say, that have a stylet. So those parasites, maybe some of your plant nematodes, they're going to burrow into plants. All right, burrow into plants. Maybe you have accidental penetration of the host. It gets into the host immune system wipes it out. It's gone. But what if you have a mutation where that worm gets in and can survive a little bit longer? Well, if it can survive a little bit longer, it actually gives it a possibility of migrating through the body to get back to the gut where it could then get out. All right, it's a much more complicated pathway because you have to get in, you have to be pretty inert or be able to fight off the host immune system, and then you have to travel through the body, through the tissue, through the body in order to get to the gut so that our worm can get out. All right, so again, if you have that situation where migration starts to become required, then you now have the possibility where maybe it's more beneficial for our worm to remain in the gut, so get to the gut and then remain in the gut, and let the eggs leave the host and then rely on the juvenile stages. Now, and that's where you can get it fixed. Now, how would this occur? What organisms? It's thought that if you're going to take this route, you're going to go into an organism that has thin skin, all right, requires some sort of moist environment, and is low to the ground, so you increase the surface area on which these worms can penetrate. Possible hosts are these early tetrapods. So early tetrapods were associated with water. They typically had thin skin. Think more of an enlarged salamander. All right, thin skin, easily to penetrate. All right, you're in moist areas because you need that moist environment. It's not because of the host itself, but because the worms can't dry out. So it's thought that these early tetrapods could have been the first hosts for parasites that exhibit this type of pattern. Now, as you're kind of seeing, these are two different patterns. You've got one where you just get consumed, and then you remain in the gut, and then you have this other one where you've got penetration, migration through the body, just to finally get to the gut. And we're going to see example life cycles of both of these types. Any questions? So the first one is, or the hookworms, is this, the penetration of the host by the worm. We're going to start with those. Reason they're called hookworms isn't because they've got hooks in their teeth or anything, but because of the shape. All right, so they've got a buccal capsule that's sclerotized. Some people would say it's because of the curled tail. Not quite. The females typically don't have curved tails. It's actually, though, when you look at them, the anterior region tends to form a kind of like a shepherd's hook, a slight bend to it. So they called it hookworms. Now, the morphology of the hookworms is similar across all of our species. I think you guys have seen this. All those different, we had Ancylostoma, Necator, we've got human Ancylostoma, duodenale, and we've got Caninum and Braziliensis. The morphology is very similar in that we have some sort of sclerotized buccal capsule. That buccal capsule is going to be armed with either cutting plates or teeth or lancets, which would be kind of like just points, all right, that they then use for feeding. So they're going to use those, slight attachment, but they're going to cut the host's epithelial tissue in the gut so that they can feed on the cells, the mucus, and the blood. All right? All hookworms have this. Our males, our hookworm males, are going to have a well-developed copulatory bursa. And again, we've seen that in the lab. All right, what's its use? To grasp and hold onto that female during mating. The eggs of hookworms are thin-walled. Right, they're susceptible to desiccation. It kind of links back to the need for a moist environment. We're going to come back to this idea. Eggs are susceptible to desiccation when we talk about the epidemiology, where we tend to see a lot of infections. Our larval stages are J1s, J2s, they're out in the soil feeding, and then the J3 stages stop feeding, and those are going to be the ones that penetrate into the host. Now, we can tell these two apart based on their esophagus. And in the lab, we have some slides that we could use to identify the esophagus types, but man, our esophagus types are really hard to see on those slides. So,
can you go back?
yep. So what I have is we're going to talk about these two different types. Main idea is, with our bacteriivores, they exhibit this type of esophagus where it's muscular. Then you have this constriction right where the nerve ring is. That constriction is called the isthmus, and then it enlarges again into almost like a complete pharyngeal bulb. Filariforms don't have that constriction, and usually they don't have as strong of muscles or as thick of a muscular esophagus at the anterior end. They still typically have an enlargement at the posterior part, but not so much throughout the rest of it. And I have a diagram ahead. All right. With our larval stages, these J3s tend to retain the J2 cuticle. So when they molt, they don't shed that J2. They hold on to it. It's possible that this is providing some sort of additional benefit, additional protection while these worms are out in the soil, or it provides some sort of additional benefit when they initially penetrate into the host. Now, I'll tell you, when they get into the host, they drop that cuticle pretty darn quickly. So I don't think it really provides a huge amount of benefit during that initial penetration stage. All right, we good? All right, life cycle. Our life cycle is going to be similar for all of our hookworms. Right. Similar pattern for all of our hookworms. There are some exceptions, and the exceptions come in terms of how the J3s can penetrate, or where they penetrate, what part of the body they penetrate. So I'm not going to diagram a specific species. We're just going to call hookworms. Just call them hookworms. Make sure I'm on board. Yeah, of course. We are low. That works. Hookworms. Yep. It's cut off just a little bit, but you can tell it's hookworms. All right, so our adult, we're going to start with our adult worm. It's going to be in our small intestine. Where it's feeding. All right, so it's going to be feeding on blood. It's going to be feeding on epithelial tissue, causing some damage, which is a possibility of pathology. All right, our eggs, our adults are going to release the eggs. Eggs are going to get released in the feces. It's an easy way to get out. All right, and then our eggs, out in the environment, out in the soils, will hatch, releasing our J1s. All right, J1s are going to commence feeding, growing, and in about 2 to 3 days, they will molt to a J2. All right, so it is temperature dependent, so the warmer it is, the faster they'll develop, faster they'll feed. All right, so in about 2 to 3 days in typical environment, they'll molt to the J4 or J2s. They will continue growing and feeding, and will molt to the J3 stage, where we may retain the J2 cuticle. May retain that J2 cuticle at that stage, but it's still a J3. All right, this is about 5 days or so. All right, now we're at the stage that is infective to our host. And our J3s now will penetrate into that host. Penetrate the skin of that host. All right, so we're out in the environment here. All right. Our host at least for us, it'll be human or dog. All right, depends on the species. There are many other hookworms out there that infect other organisms. All right, but for our species, we're going to say it's either hookworm or dog. All right, so we're going to penetrate, and at this step, when we penetrate, our cuticle gets shed. So if that J2 cuticle has remained, it gets shed shortly after it penetrates into the host. So now we're inside the host. That J3 is going to get into the circulatory system or the lymphatic system. So remember, our goal is to get to the small intestine. That's where we need to get to. So there's a couple different ways we can do that. There are some worms that actually penetrate and they get to the intestine the shortest way possible. Start just kind of burrowing through tissue until they find it. These guys are going to get into the circulatory or lymphatic system. Right? They're going to utilize that system to get to the heart. So they are still in blood or the lymph. If they're in the lymphatic system, they get to the heart and they ultimately get to the blood into the circulatory system. From the heart, they then take the right side of the heart and go to the lungs, where our J3s are going to burst out of the circulatory system. All right, they're going to burst out of the circulatory system at the alveoli of the lungs. So what's surrounding those alveoli? So what are the alveoli? Alveolus. But resident human anatomist.
Isn't it like break?
Yeah, so your lungs gets down into often in diagrams like circles, right, where the blood capillaries are. Yeah, those are the alveoli. All right, so it's going to burst out of the capillaries into the alveoli of the lungs. Again, cytopathology. Now, once we get into the lungs, we will get carried up to the glottis by ciliary action. All right, so inside the lungs, we've got some cilia that will move. Cilia help to move particles out. All right, it's going to move our worms, our J3s, up to the glottis, where we will then cough and swallow. So it gets us up there. We got something. Clear our throat. Get some phlegm from the lungs up. We swallow it. Unbeknownst to you, you just swallowed a bunch of J3s. J3s are now in the small intestine. All right, once we get there, that J3 finishes, it molts to our J4, molts to the adult stage, where it then finds a mate, mates, and starts reproducing. All right, egg production commences about 5 weeks post-infection. About 5 weeks post-infection. When you first get penetrated by these J3s, about 5 weeks later is when you start producing eggs. Now, there are exceptions. We'll talk about those. That's generally speaking. All right, so some things about the life cycle. All of this step here, all right, we're out in the environment. All right, out here we are susceptible to desiccation. Susceptible to desiccation and freezing. All right, so kind of a reason why you have lower hookworm prevalences in far north or the extreme south, southern hemisphere, how about the poles? Okay, say the poles. It's susceptible to freezing. The desert-like environment is susceptible to desiccation. It needs some moisture. Our development is also temperature dependent. So it develops faster in warmer areas, slower in cooler areas. And it's part of the reason why this is more of tropical, subtropical parasite. It needs the warmth. These J1s, J2s are going to feed on bacteria and organic matter. Organic matter that typically accompanies the feces. All right. Now, how do the J3s know when to start penetrating? These things sense warmth, so rapid increase in temperature, and fatty acids. So the warmth kind of gets them moving, gets them thrashing about, and then once they contact with the skin, they sense the fatty acids, and they start initiating the penetration behavior. Doesn't do you good if all of a sudden they sense, let's say, a leaf that falls on the ground. Why try to penetrate that? If you penetrate it, you're going to drop your cuticle and make you even more susceptible to desiccation. So you need both of these triggers. Fatty acid. And again, it's behaviors. They're programmed behaviors that when they detect the cues, they're now going to start moving, thrashing, and trying to penetrate into the host. Now, how do they penetrate into the host? Depends on the species. Some of them will secrete some enzymes that you have an enzymatic entry. Others will seek out areas between the cells or at the base of the hair follicles and will try to wiggle their way through those cell junctions. All right, so it just kind of depends on the species that we're talking about. Any questions on hookworms? Right. So variations to this, exceptions, comes down to where they penetrate. Comes down to where they penetrate. All right, so human hookworms can penetrate through the skin, or they can penetrate through the mucosa. All right, so if you put dirt in your mouth that has a J1, J3s, those worms can penetrate the oral mucosa, enter the circulatory or the lymphatic system, undergo the migration to get to the intestines. All right. Most likely places where these things penetrate are going to be the feet, the hands, and the buttocks. Why the buttocks? If I tell you that that is most common in young children, does that give you an idea? By young children, I'm not talking about like 10, 11-year-old kids. I'm talking like 1, 2, maybe 3-year-old kids who aren't potty trained yet. Well, yeah, they'll take their diapers off, and then kids do that, run around, and sit on the ground, right? Laughing and think nothing of it. All right, so human hookworms goes through the skin, goes through the oral mucosa. Dog hookworms, however, have an alternative route that they can take. So they can penetrate through the skin, they can penetrate through the oral mucosa, but some of our dog hookworms can also be swallowed. So if the J3 actually gets swallowed, oftentimes those J3s skip the migration and they just start developing to the J4 in the adults. All right. We also have the possibility of 2 other types of transmission pathways. We've got transmammary transmission and transplacental transmission. These are known to occur in the dog hookworms, probably occurs in other species. I'm not sure if we know about humans. Maybe it's suggested that humans can have this. It's just we're in a pretty clean environment. So what is this? Transmammary transmission is transfer of the larval stages through the milk. All right, so this isn't going to occur when you have an adult. The adult's not going to migrate. This is when you're out, you're pregnant, or you have your kids, breastfeeding them. You get nailed by a J3. It's undergoing the circulation, and it takes the circulation, gets into the breast milk, and then gets transferred to the child. All right. Same thing could happen with pregnant dogs. We'll keep dogs. Pregnant dogs. All right. They get nailed, J3's penetrating, gets into the circulatory system, and then goes through the placental to get into that developing fetus, to the newborn. So it is possible that we have both types of transmission. So transmammary is through the milk. Transplacental would be the sharing of the blood, with the placenta, umbilical cords, and so forth. All right, so our larval stages have a J1, the larval stage, J1, J2, have a rhabditiform esophagus. This is typical of bacteriivore nematodes. And again, there's numerous nematodes. We can go out to moist environments, collect soil, and probably find, numerous species of these nematodes. This is an example of a rhabditiform esophagus. All right, it's actually has different parts of it. It's muscular. Right, just across the entire range. But we've got the anterior muscular part with a slight enlargement, a narrowing, and then an enlargement right near our intestinal junction. All right, and they've got different names for this. So this part here is called the procorpus. Procorpus, basically the first part of the esophagus. The slight bulge that it develops, that's called the metacorpus. And then the narrowing is the isthmus. And that narrowing is where we would find the nerve ring. And then we've got enlargement for the bulb, esophageal bulb or pharyngeal bulb. Don't have a J3. So that's typical of our J1, J2s. J3s, they have a Filariform esophagus type. All right, this esophagus type doesn't have a metacorpus, doesn't have an isthmus. So what it basically is this procorpus extends the length to the pharyngeal bulb. And even the pharyngeal bulb might not be super prominent. You just see that it gets larger. All right. So I do think if you do search for Filariform esophagus or Rhabditiform esophagus, there have been plenty of invertebrate classes, zoology classes, parasitology classes that have posted their drawings. So you could check out those. But big difference, procorpus, metacorpus, isthmus, and the bulb for Rhabditiform esophagus. For Filariform, it is just basically procorpus and the bulb. And there's really no constriction for that nerve root. You don't see it. Ready? All right, so there's two human hookworms that we're going to talk about. Necator americanus and Ancylosma duodenale. All right, we've got Necator species, we have Ancylosma species. All right, there are differences between the two. We could identify differences, but we're not going to in the lab. So in the lab, it would just be what is the species? Hookworm is sufficient. All right. Differences come in their bursa and in their buccal capsules. So you can see that Ancylosma has these teeth. All right, they have these teeth, and I believe Ancylosma brasiliensis, the sides that we have, I think most, if not all, of those worms are on the side, and you can make out a tooth that's sticking out, or maybe two teeth sticking out. Necator has cutting plates. So it's instead of having like teeth, it's got these rounded edges that are say plates. They're kind of like the slicers at the deli counter, right? That's kind of what they use for. They can kind of cut. They're sharp and they can cut. They do have some papilla associated with it, but plates, not teeth. In terms of the bursa, Necator tends to be much wider, whereas Ancylosma is much more circular. So again, if you think about our slides, there's going to be no way to really turn the slides on edge so you can see, well, is this circular or is this more rounded? And of course, pattern of our rays, bursal rays, that also has something to do with it. Hey, but again, I'm not going to ask for these differences. But which one has cutting teeth? Which one has cutting plates? No, we're not going to talk about. The reason we talk about these two is because they have some other differences, not just in their morphology. So Necator, skin penetration only. All right, it has to go through the skin, the hand, the feet, the leg, whatever. Ancylosma can penetrate the skin or can penetrate the oral mucosa. All right, so if you put dirt in that's contaminated into your mouth, that's contaminated with Necator, you won't get it. All right, it needs to go through the skin. I mean, I guess you could. It penetrates through your hands. Lifespan of these things are also different. So Necator has a 3 to 5 year lifespan. Once they get to the gut, they're going to live for 3 to 5 years releasing eggs. Ancylosma only lives for about a year. All right, so if you're going to get infected and if you're going to retain this infection, you have to be reinfected basically every year. This difference in the years likely is the reason why we see arrested development in Ancylosma, but not Necator. All right, so the arrested development is going to happen when our J3s penetrate. They basically stop. They stop migrating. They stop maturing or developing. They just hang out in the body and wait. Wait until they were, whatever their programming says, to start coming out and undergo the migration again. All right, so this arrested development is probably tied to the lifespan. It's also tied to the environment. You guys ready? So what is this arrested development? Well, these worms are going to stop developing for up to 38 weeks. And they're going to enter the host. They're going to just stop. At this time, they are being hit by the host immune system. All right? But their cuticle is very, very resistant to any sort of damage from the host immune system. So they just hang out. They don't have to hide inside a cell. They don't have to do immune suppression. They don't even have to take up host molecules and express it. Their cuticle is pretty inert. It's not going to be harmed. So these things penetrate into the host, and then they just stop. They don't enter the lymphatic system. They don't enter the circulatory system. They just remain in whatever tissue they initially penetrated. And then at some random time, in that up to 30-week time period, all of a sudden they come out of their arrested development, enter the blood or the lymphatic system, and undergo their migration or complete their migration. The trigger for these guys is the onset of the dry season. All right, so the drying out of the soils will now start to predispose these developing juvenile stages so that more and more of them will undergo this developmental arrest. All right, so why do that? Well, why they do that, you have to think about where they live, where we typically find them, and the lifespan of our worm. So that's what these diagrams are here to kind of show you. So let's say we had no arrested development whatsoever. We've got wet seasons and dry seasons. So this dotted part is the wet season, conducive for our juveniles to mature, develop in the soils. Right? And then we have the dry season where if an egg gets released, if it hatches, the juveniles dry out, they desiccate, and that's it. All right. In those areas, it's seasonal. So you've got seasonal rains, seasonal dry season. Because we're talking like Southeast Asia. I think, Cambodia, Vietnam type areas. You've got your monsoon seasons and your dry seasons and so forth. All right. So our worm lives about a year. All right. Lives about a year. So I have it spaced out here to where we have our larval stages in red and our adults inside the host in black. So if an egg enters the environment during the wet season, the eggs can develop, juveniles can develop, it can penetrate into the host and be successful. If it comes out, let's say in the middle part of our wet season, same thing, we've got plenty of time to develop and find another host. But if you get released right at the tail end of that wet season or at the start of the dry season, those worms aren't going to make it. They don't develop. So you're going to lose all of those guys. You're going to lose all of these individuals, all of these individuals. And this is what we're saying is we're representing different times when the eggs get laid. Now, these worms only live a year. So this first worm, it started producing eggs. You can see it started producing eggs at this time. So this is like, I've lined up the lines at about that 5-week mark maybe, not to scale. Right? But this worm starts producing eggs, and it can get a next generation into the host. But once we get to that dry season, the rest of those eggs that this worm produces, they're lost. On this second generation, most of these eggs during the first part of its lifespan, they're lost. But that later stage, it can get to the next host. So overall, if we had a life cycle like this, you're running a risk that your host gets infected right near the start of our dry season, and then none or very few of our offspring ever make it in. With this developmental arrest, we have it programmed so that some of these worms that get into it, they wait. And they come out at a random time. So some of them undergo developmental arrest for a week. Those guys, very short dashed line up here. So this is development in the soils, and then dashed line here is our developmental arrest. They're not developing. And then when it becomes solid, they undergo their migration and complete it. Some of them come out very quickly. So those, hey, they're producing eggs. Others remain in developmental arrest and come out perhaps in the dry season. If you come out later in the dry season, you still have most of the wet season where you can produce your eggs. And then it continues on. These guys, they came out, they infected middle of the dry season. Or these eggs are lost. You could say these eggs were lost when they got dropped. So I should have put an X. I think it did have an X. I didn't group it appropriately. But we lose this guy. This guy's fine. This guy's probably lost. This guy's fine. So what we're doing is we're staggering when our eggs go out in the environment. We're doing it in kind of a random fashion. And by doing that, we avoid this repetitive pattern of wet season, dry season, wet season, and dry season. And again, these worms only live for a year. So by doing this, by delaying, when we have the worms coming out, some of these are long, so Some of the lines are long, some of the lines are short. We're trying to make sure that our infection intensities remain fairly high to ensure that the next generation of hookworms can get into that next host. All right? Hookworms aren't the only organism that do something like this. All right? The whole reason is to kind of buffer that uncertainty of the environment. All right? Think about seeds of flowering plants. If you take a batch of seeds and you try to germinate them, what percentage of seeds actually germinate? How many of you have ever done that? In like a high school class or a college class? You know?
Yeah, that's true. You call it yeah.
Did we talk about seed?
Mhm.
Sure.
Or
Oh
like
yeah, the
earth.
grass seeds. Yeah. Yeah, we did.
Yeah, in regards to like the water from the plants.
Yep. Yeah, so we didn't have 100%. Sometimes we do. They do have selection, but just on a regular flowering plant in a year, right? They overwinter and then spring comes around. If all 100% of the seeds germinated, what happens if that's a bad year?
You're gonna lose all of it.
You lose all of them. So one of the strategies is instead of germinating 100%, there's some genetics that'll say, hey, instead of waiting one freeze-thaw period, I need to wait 2 or 3, in the hope that at least some of your seeds germinate during a good time period. So plants do this with the seeds. We have some marsupials. Kangaroos do this, believe it or not. They always have an offspring with them. And it's the same philosophy. If they lose one, they have another one like in the wing to balance this uncertainty. So yeah, with our nematodes, it's all this developmental arrest is all about increasing the chances that our egg production of that worm will coincide with favorable conditions. But it's not just the individual. We have to think about the family of those worms, the genotypes of those worms, so that our population remains stable. Ancylosma duodenale needs this. It's 1 year. If you have a worm that lives 3 to 5 years, like Nicator, hey, you've got multiple periods where that worm is going to be dropping eggs and its offspring can survive. So you don't really need to do this developmental arrest. All right. So where do we see this sort of prevalence of hookworms? We typically have it correlated. So high prevalence is percentage of individuals that are infected. Intensity is the number of worms per infected individual. Right? It's going to be correlated with environmental conditions and with night soil. So with environmental conditions, we need loose aerated soils. Right? We need soils where oxygen can get down because our worms are going to utilize oxygen for ATP production. The warmer environments definitely speed up development. All right? And the moisture helps to reduce desiccation. All right? So just loose, aerated, moist, warm soils, that's what we're looking for. And those are like optimal conditions. So what types of habitats are these? Well, the ideal places tend to be wooded or forested habitats. Why wooded or forested habitats? Well, big part of it is the shade. You cast a shade down. You don't. If the shade is present, you don't dry the soil out as quickly. And in a lot of those areas, you get regular rainfall. So the southeastern United States, very popular one. Very popular environment. So much so that we still have hookworm problems down there. Who am I going to be? I forgot one. Forgot an image. I meant to put it up. We're almost done here for today. Jeez, that one. Crazy what Google searches are giving me right now.
Mm-hmm.
All right. Well, anyways, down in Southeast United States, as recently as like the 70s and 80s, 1970s, 1980s, they'd have a billboard campaign up. And now we're talking about Southeastern United States, a very poor area, where you don't have like indoor sewage, indoor plumbing, so they're still using outhouses. One of the campaigns is they had a big hole in the billboard and a small hole in the billboard, and it just said "cut one for the kids." Because what they wanted to do was to encourage the kids to use these outhouses to keep them from defecating out just out in the soils around their area, around their home. Because if they did that, a lot of those kids, a lot of the adults are walking around barefoot, not with shoes. So their foot's constantly in contact with the ground. What they're trying to do is try to contain where those juvenile stages are developing in the hope that we disrupt transmission. All right, so southeastern United States had hookworm problems. The other association is with night soil, so using human excrement as fertilizer. So this was a picture from the BBC.org where they did a article, and it was a relatively recent article on night soil collectors in Bangalore. So India, Bangladesh regions. Still happens. So they're collecting feces, they're using it out as fertilizer. And those people that, if you think about rice fields, right, they're using it as fertilizer, and you have a lot of people out in the mud and muck, in the water, harvesting, taking care of them. They are exposed to those juveniles. So a lot of these places, some of the genetics really shows that you have these family units anytime they're using night soils and farming their own land. So, and that's in the parasites. So the parasites themselves form these subpopulations that were tied to family units. Questions? All right, so I think what we'll do is we'll stop with the pathology. We'll pick up the pathology on Monday, and then we will move let's see how much more. Oh yeah. Yeah, we'll finish the pathology on Monday, and then probably start with our ascaris. So the second part will go Monday, Wednesday, and then probably Wednesday, Friday for our filarial ones. All right, that's good. If you guys have questions, please email me.